We were able to squeeze this into Nature at the end of last year. For anyone who's particularly inspired by this talk, it actually reaches paper tomorrow, which is quite timely. I know what a lot of my relatives might be getting for Christmas this year. And it concerns the timing and the development of eukaryotes. 2 QR codes which sort of link the obvious. The talk's going to break down into 3 sections, a relatively brief introduction about why this is a question, a rather large middle section on the method, because the method is actually rather complicated. It has a lot of power. We think we could be applied to quite a few other different questions, especially where we have development of lots of features or long branch lengths and that kind of thing. And the final part is basically the results. But the first half of that results is trying to convince you that we're actually getting signal and information at all. And then we round off with how we actually think what the developmental timeline for eukaryotes actually looks like. So I must admit I approached the School of Astronomy with trepidation. I have to explain the difference between prokaryotes and eukaryotes. We've done a lot of public outreach, and so what I tend to lean on is that eukaryotes are what we characterize as complex life. That includes things like myself, my students, other animals, plants, fungi, but extending into the microscopic, such as algae, yeasts, and amoeba. They are distinct from simpler forms of life in the sense that they have many compartments. And beyond the microscopic of just having just more compartments, they often possess rather sophisticated and complex molecular interaction systems. Their genomes tend to be more elaborate with more levels of layers of control. And so those are the eukaryotes, which we'll describe all complex life as eukaryotes from this point on, and the prokaryotes, which encompass the domains of bacteria and archaea. By contrast, these are more simply organized systems. I don't like saying they're simple because, these are still evolutionary machines which are very sophisticated in their own way, but they certainly don't have the complex internal architecture or perhaps the complex history that eukaryotes do. But we have a dividing line down the middle of this slide with sort of just 2 camps. And we look at a eukaryotic cell and it's packed with complex features which we can't find in prokaryotes. We have fairly good evidence that prokaryotes are the older domains of life. And so at some point, we must have this transition from a simply organized cell to one with all of this intracellular complexity. So there has to have been a period where this complexity developed and these various features emerged. But to date, we don't have any living intermediates for halfway between this, which sets a problem for us phylogenetically. The ideal situation for phylogeneticists like myself, am I finally going to describe myself as a phylogeneticist? Maybe. The ideal situation is that you have a number of intermediate states which possess some of the features. You then build a tree, and your tree will tell you which things occurred in which order. Unfortunately, with the transition from prokaryotes to eukaryotes, we have one side with none of the features and the other side with all of the features. So traditional phylogenetics can't work out the order of this feature acquisition. So I've said there's no living lineages which represent an intermediate state. What do we have in terms of fossil evidence? Well, I do get a reputation of being quite hard on paleontologists. It's not intended. It's just I want answers in this middle region here. So we have fairly good evidence for ancient bacteria going back more than 3 billion years. A paper by our group, I think about 2 or 3 years ago now, suggests that Luca, the last universal common ancestor, is about 4.3 billion years old. We can identify fossil eukaryotes which we're pretty certain belong to groups that we recognize. And we've got a whole bunch of other things of certainly a eukaryote grade organism, but which we can't confidently assign to any extant taxa. And so we do have some information from the fossil record. We can certainly set a minimum age for the last eukaryotic common ancestor, but the fossil record doesn't really distinguish between extinct or stem eukaryotes. It doesn't seem to preserve the subcellular detail that we want to fully populate our list of developed eukaryotic features. As I say, I'm very hard on the paleontologists, but other work done by molecular biologists, in particular building time-resolved trees of life, has similar kind of impact. We have a range of estimated dates for the last eukaryotic common ancestor, which themselves span hundreds of millions of years across and are uncertain by an order of 1 billion years, give or take. And so, taken together, the evidence that we have from fossil record and previous data that we have from molecular clock analyses, we're still fairly uncertain about the eukaryogenesis timeline, with plus or minus 2 billion years on a planet 4 billion years old. Worse, in terms of molecular biology and building trees of life, a tree of life will only connect eukaryotes to prokaryotes along a long line, and that line doesn't actually tell us about the development of any features. It just connects 2 points. It will give us the timeline, but it won't give us the development of features. Other evidence from molecular biology has, especially in the more recent era where we can look at genes and we can sequence things and this kind of thing, tells us that compartments of the eukaryotic cell appear to have different prokaryotic origins. And so I think going back to sort of the Sagan era in the 60s, we had a fairly good idea that things like the mitochondria, for example, were at one point free living alpha proteobacteria. We can look at other gene families in other parts of the cell, and we can see they are clearly archaea. It gives us an unusual situation where this isn't a species which has progressively just acquired features. It's actually a chimeric organism which has been the input of more than one prokaryotic lineage. That's great. We have an event. We know that somehow archaea plus alpha proteobacteria eventually leads to eukaryotes, but it doesn't tell us at what point they met. I mean, did that meeting between archaea and mitochondria happen at the beginning, or was this the last process? And where are features related to this A plus B? And so just knowing where things come from isn't sufficient to work out a timeline of eukaryotic feature development. And because of this, because we have a relatively large degree of uncertainty about the time of eukaryotes, we know that alpha proteobacteria and Asgard archaea somehow equal eukaryote in the end, but we don't know the order. And so over the last 50 or 60 years, we've been accumulating hypotheses which we're unable to disprove one way or the other, where some people reason that the mitochondria was first, some people reason mitochondria was last, some people invoke a number of other organisms involved in eukaryogenesis as well, based on molecular phylogeny. And we still don't have a concrete solution one way or the other. We just have a lot of hypotheses. And yeah, to summarize the introductory section, we have a very poor understanding about the length of time it took, or even when the last eukaryotic common ancestor was. It's a rather bleak situation because we don't have fossils and we're not likely to get fossils which encode the intermediate states. But we do at least have some evidence, which is the fossils can at least tell us some of the maximal and minimal ages of certain groups. We know there are formative events, though these themselves don't actually contribute to understanding how the features developed. But we do have a relatively mature understanding about which genes belong to which compartment and feature in eukaryotes. That's useful. And so, again, I took up this postdoc with some trepidation. The solution to eukaryogenesis in 18 months was a steep ask, but I was also needing the money. And we decided to try and tackle this by doing two things. First, it'd be really nice to be slightly better than plus or minus a billion years. So we wanted an accurate timeline. The second thing is there some way where we can look at the gene families associated with features and figure out when these features occurred? And for the remainder of the talk, we're going to look at how we developed this approach and then how we tested it later. But it basically boils down to two parts: building this tree, trying to extract data from single gene families. And so sort of onto the second segment now, how we actually did it. So this is a slight rearrangement of that tree before. The tree before is one that I do sort of public-facing graphics kind of thing. Oh, look, two things come together in eukaryotes. But if we consider LICA, for example, any individual gene in LICA will either have been invented along the eukaryotic stem, or it will have been inherited from alpha proteobacteria, or it will have been inherited from Asgard archaea, or perhaps that gene was present in the last eukaryotic common ancestor, and eukaryotes actually get two copies. But This gives you an unusual situation because if you pursue that kind of logic, then eukaryotes are a set of genes. In fact, it's a set of sets of genes. And so there are a set of genes in LCLU which descend from Proteobacteria, and there's a set of genes that descend from Asgard archaea. We can kind of exploit this in a way because the node which corresponds to the set of genes inherited from alpha proteobacteria at LCLU must be the same age as the set of genes speciating at LCLU from Asgard archaea. And one can imagine that down here in the corner there's a split between mice and men, which is actually mirrored by a split in the top corner, which is mirrored by mice and men. So the ages of these nodes have to be equal. And so this is an approach called cross-bracing. It's quite useful where we don't have all too many fossil calibrations deep in the tree here, but we can feed temporal information from these LCLU clades. And so the actual building of the tree is kind of mundane. I don't really want to bore everyone with how to do it, but it basically involved identifying groups of genes which descend from alpha proteobacteria, groups of genes that descend from Asgard archaea, groups of genes present in eukaryotic common ancestor. Every single eukaryotic species appears at least twice in this tree. And in fact, this little subtree is identically mirrored at 2 points in the tree, and every single subnode of that tree is actually cross-braced as well. It is decorated with the fossil data and calibrations that we do have, but it's not oh so many. A lot of our calibrations are coming from eukaryotes, I have to admit. In terms of the size of a tree, we're talking 300-400 species, but with the fact that you have to double this number at very least. And so we went ahead and we built this, we gathered our genes, and we managed to create a supplementary figure 1, which looks an awful lot like this diagram I've presented here. But this is quite interesting in itself. It gives some ages from the deep nodes. The cross-bracing effect, where we've linked nodes of the same age, is quite apparent because if you see where LCLU is here and LCLU is here, they're identical in age. And actually, if you have fairly good eyesight, you'll see that all of the subtrees of eukaryotes also share the same age. We obviously have some impression of the timeline of eukaryogenesis. It's that. But the immediately striking thing that we had when we actually visualized this tree is that the genes descending from archaea seem to split off a lot further back in time than the genes that descend from alpha proteobacteria. So the stem lengths to eukaryotes are actually quite substantially different. There we go, there's another summary of it. Although we know the timeline, and we can say things like all eukaryotic features that descend from alpha proteobacteria have to have occurred in this time window. Now, that's fine. We know those features have to occur in this interval here. But if we have a feature descending from archaea, does the feature develop before we even have the mitochondrial endosymbiosis? Does it develop at mitochondrial endosymbiosis? Does it happen later? The species tree sets the timeline, but we can't see where the development is happening just from the species tree. Then we have to sort of think a little bit more hypothetically. And so if we have a feature in eukaryotes or a function in eukaryotes, and we find an ortholog in prokaryotes, which does exactly the same thing, we can assume that feature or function was inherited from that prokaryotic group. That's fine. But this isn't a developmental process, this is it inheriting a function. This is the big conceptual leap and the whole thing that the paper's about that made it to the title. If we have a gene that actually undergoes a duplication on the stem, and one of the paralogs actually develops a new function, we can actually start to set up a logic whereby we can set a maximal age for the development of this function, all things being equal, some assumptions made. But it means that if we can, and of course, this tree has a whole extra node versus the species tree. And so if we can associate these additional nodes with the acquisition of features or functions, then we should be able to place them on the eukaryogenesis timeline. The other question is, we're asking a quite big ask. we're asking for genes to have a very particular signal. They must descend from either alpha proteobacteria or Asgard, and they must undergo a duplication on the stem. Do these things actually exist? The answer is yes, but actually finding them is actually a little tricky. And so these events certainly exist. And again, all the coloration in this presentation is sort of the same throughout. We have archaea here, there's a pre-Leaker duplication, and we have 2 Leaker radiations. But just finding them like that is rare. Often what we're dealing with are gene families which actually go all the way back to Luca, and we have these rather complicated evolutionary scenarios. So when we look through the history of a gene family, we often have this rather more complicated picture. We can see something that we kind of like to analyse there, and it was very difficult developing an automated approach to try and extract these pre-Leaker duplications. Because I'm quite proud of this, I will give it a minute or two in this presentation, but it's a nice approach, I quite like it. And so here we have that gene family that goes probably back to Luca. We segment the tree coarsely, and we kind of count what's in each segment. So this one is red, which means it's archaea by my code, and we can produce a little summary tree with only a few branches with what those branches contain. And so green in this case means eukaryotes. If we take a reference species, I think for our paper we mostly use either yeast or Homo sapiens, we can see that in this big tree we can find 3 reference sequences, which I've labelled A, B and C. What we can then do is we can then just apply some sort of algorithmic logic. We can say for A, traverse the little tree until you reach a group which contains prokaryotes. And where you have 2 reference sequences traversing the tree to the same point, then you've probably got a good candidate for a pre-Leaker duplication. Using this approach, we managed to extract the majority of our pre-Leaker duplications. Actually, just finding them as nice little triads is actually very difficult to achieve. But using this approach, we managed to find 150 gene families which were amenable to analysis. We were quite conservative. We wanted our trees to be fairly well resolved, to have unambiguous Leaker nodes. The actual number of genes that exhibit this pattern might be quite high. Other publications, like the Trier and Vosberg papers, suggest there might be hundreds. We were happy with this 150 well-separated, easily explained, transparently pre-Leaker duplications. But the caveat here is, although we have all of these events that potentially occur on the eukaryotic stem, the single gene trees themselves don't actually contain much information. We're talking about single gene trees, we're talking about proteins, we're talking alignments of, at best, hundreds of amino acids. The actual amount of information they encode is relatively little. The ability for them to structure single gene trees is fairly limited. But what we do have is that time-resolved species tree, which leverages dozens and dozens of gene families, which has got an alignment of tens of thousands of amino acids long. But we can use that species tree to reconcile and root the single gene trees. So we used a program called TreeRex. I use ALRAX. There's a number of different ways you can achieve this. I won't dwell on it particularly. But we can certainly root the single gene trees, and we can then, using our species tree as a guide, apply temporal information to the single gene tree. And when I say apply temporal information, apply priors from the species tree to the single gene tree, we mean we add a lot of priors to the single gene tree. And so here we have that rooted single gene tree. It's been reconciled and rooted. And each one of these dots is some information about the timing of that speciation node we got from the species tree. So these things are densely annotated from the species tree. We also use the same trick. In the bottom corner, we have the split between mice and men, and we know that has to be the same age on both sides of that tree. So we can also cross-link them as well, or cross-brace them, sorry. By and large, the tree is so densely annotated that in terms of our deep nodes, the gene duplication is probably one of the few nodes which isn't annotated with some sort of temporal information. We can then time resolve these trees, and we do get an age distribution for that duplication node. Notice the width is actually quite wide compared to some of our confidence intervals for the ends of the eukaryogenesis timeline, but this seems to be the sort of plus or minus 100 million years seems to be pretty good for this kind of thing. Anyway, just to round off that section, we built a time-resolved tree. We have a method that can date these internal duplications, which may lead us into a way of defining how features developed. We have a lot of them. So it's 150 gene families, but some of these gene families undergo multiple rounds of duplication, or they possess multiple separate instances of pre-leaker duplication. This is figure 4. We've got sheets and sheets of these. It took me better part of a month just to draw this damn paper, but we have a lot of duplications. But what can we extract from them? Well, as I promised, I was going to why should you believe me section of the results. And so if we consider the timeline that we're proposing, how does that sit with previously published studies? And so the width of our intervals is quite short. They're not the shortest, but it's quite short. There's another attempt to produce a time-resolved tree of life by Mahendra Raja, who I met at Royal Society beginning of last year. Very nice chap. And we both use this cross-bracing approach. It seems that the cross-bracing approach is probably the reason we get these quite tight intervals. But it's worth noting that this might be a useful method if it works out. Our age for leaker overlaps with lots of other people's age for leaker, so I don't feel that's particularly controversial. Our age for the split from alpha proteobacteria does overlap other people, but again, in terms of a consensus of information, it doesn't stand out, but we're still not very confident about that. In terms of our end fecal date, again, it overlaps previous studies, but we are towards the older end of this. So superficially looking at our dates, we feel that we are overlapping other studies. We have some confidence that our numbers aren't entirely crazy, but because the timeline is so important to us, because we apply this to the single gene trees, we decided to do some additional sensitivity analyses to like really make sure our timeline is robust. And so we basically took the same alignment, but we tried it with different calibrations and different tree topologies. And what we found is that if we change the position of the eukaryotic root, which is still, unsolved, we could change the age of leaker by plus or minus 100 million years. Not really big changes. And it doesn't seem to change the age of any of the splits from prokaryotes. Again, there's some debate about where exactly eukaryotes split from the Asgard archaea. And again, we tried a number of different positions within the Asgard archaea, and we found that you can change the end fecal age a little bit, but actually, well, the end fecal age is probably not going to be affected anyway, but leaker age basically stays the same. In terms of our sort of uncertainty about deeper parts of the tree, the deeper we go, the less kind of confident we are in our results. And we tried setting some variations deep in the tree. And the general effect is that events very deep in the tree don't really change the dates closer to the tips. And so changing the relative ages of the last bacterial common ancestor or the last archaeal common ancestor don't really impact our timeline. And so we did it maybe, I think, 8 or 9 different ways, but we found that our timeline was fairly robust, as in plus or minus 100 million years on a billion-year process. So we're kind of happy with that. That's robust. This does have some implications because, if you remember in that introductory slide, we're fairly certain that we have eukaryotes up to 1 billion years, then we have all of these other things. But given that we think leaker is here, it probably means that these other fossil things are actually crown or very end of stem eukaryotes. And actually, we haven't started collecting fossils for what would be the intermediate state of eukaryotic development. So I don't know how pessimistic or optimistic you want to be about that, or whether they are out there to find, but we don't think we probably collected them yet. The other thing I kind of want to convince you is the single gene trees, which, as you saw, we very densely annotated. And so what I tried to show in this figure, which I kind of clumsily put together last week, is I took all of the duplications that we have, which have this basic pattern. There's a split from either proteobacteria or Asgard archaea, a single duplication followed by 2 leaker nodes. So it's the same tree topology each time. I've arranged as many as I could fit onto whatever height of thing that is. And the sort of null hypothesis would be that if there's no information, if it's driven entirely by the topology of the tree, then we'd expect those node ages to probably sit in the middle of the plot, which I've done with a coloured line, and they should probably have a symmetric distribution. What we actually have is, at least for this category of topology at least, is there's quite a large variation in age distributions and intervals. Some I don't like very much, but I felt I had to include for fairness. The width of the interval seems to be related to the length of the gene and therefore the amount of information. So this approach works best when the gene's actually quite long. And so tiny little ones like that will probably be in the order of about 500 or 600 amino acids, whereas that's going to be invariably some ribosomal protein, which I feel a bit dodgy about, but that's fine. So the first impression is that we are dealing with a population which does actually have quite a lot of complexity to it. But the point that I think that sets apart our study from other duplication studies, there has been a few recently, is that I started off as a biochemist, which means I'm actually happier thinking about genes in terms of coloured rectangles and shapes and how things interact with each other. So that's my foundation. And I realised that if there's a protein complex which contains the products of two different gene families, and at some point in evolution that complex was to duplicate between different compartments or duplicate to do novel functions, then I would expect that on those sort of complex duplication events, I would expect to see a synchronous duplication of their constituent gene families. And we observe this. It's quite nice. I mean, I don't think I've chosen the best example there. Here we have a complex which undergoes 2 rounds of duplication and its constituent gene family is going to go 2 rounds of duplication. The premise would be that the shapes would line up. I must admit the circle's not quite good, but we started looking at overlapping confidence intervals. And we have maybe 24 or so protein families in the paper which have this kind of sort of predicted duplication pattern. Here's another example where the 2 little shapes line up almost exactly, so I had to include it. But here we have a complex which underwent heteromerization and then duplication of that heteromeric complex. And again, they both line up almost identically in time. So we have a feeling from how proteins associate intermolecular complexes and how these complexes are duplicated between compartments that maybe gene duplication dates are giving us real information. The other way in which we like to sort of test this information is that if you're a compartment, sorry, this is a very interesting hypothetical thing to think about, but if you are a compartment in a eukaryote, you need proteins which target your proteome to you. You need proteins which maintain the cellular membrane. You need proteins which fuse you to other compartments or traffic proteins to you from other compartments. You need transporters. So each time you create a novel compartment which does its own thing, you're going to need custom trafficking and targeting proteins for that new compartment. So as soon as you specify a new compartment, you're sort of creating a mandate for those gene families to undergo duplication to pattern this new compartment. And so here we have an example where, again, the shapes don't line up too well in some places. Those pentagons don't look great, but again, whatever. We have a protein which undergoes duplication and it forms a heteromeric complex. It duplicates again, and we end up with a leaker node on each of those branches. In this case, the leaker nodes are patterned to the endoplasmic reticulum or the Golgi, so red and orange. We undergo another round of duplication, and what do you know? These ones are targeted to a new compartment. This is Golgi in the endolysosomal compartment. And so when you create a new compartment, you expect that there will be a synchronous duplication and patterning of this new compartment amongst multiple gene families. And so we have a number of proteins that are involved in trafficking and fusing fusing compartments to other compartments. And we can see that the very first that agree on the production of the endosome, they all kind of marry up. And so when you create a new compartment, you do seem to get this synchronous signal of patterning that compartment across completely separate gene families. These are gene families which are completely separate trees, but the duplication dates seem to agree. So this is another line of evidence to saying that these age distributions we're getting from these single gene trees aren't completely arbitrary. They are actually agreeing on evolutionary events in time. Anyway, these two sort of lines of evidence are what we have for sort of proving these gene duplications aren't just made up distributions. So I kind of hope by this point I've convinced you that the tree is okay and we've done all the necessary tests. We're confident we've done all the necessary tests that we can think of to say our timeline is correct, particularly that difference in stem lengths. We can't seem to modify our trees such that we can make them equal, except by setting them equal. We also feel that the age distributions we're getting for these single gene trees are meaningful. They do seem to behave in the expected way. And this is from this patterning of compartments and duplication of protein complexes. And you've bared with me till 35 minutes now, and now I'm going to deliver in the last 10 minutes how I think eukaryogenesis went, based on this information, if we all agree that it feels possibly correct. And so, sorry, I'll wind it back. Generally, in the eukaryogenesis argument about how eukaryotes came to be, people really care about 4 major features. They care about cytoskeleton, which is the ability of the cell to pattern extrusions or to shape its membrane, the nucleus and its genomic organization, the endomembrane system in the mitochondria. And I'm going to try and give you my gut feeling for where these things sit on the timeline. So in terms of the cytoskeleton, a lot of the duplications happen early. We would feel quite proud of that, except there's a number of different papers out there that say, at least in the Asgard archaea, that actually the cytoskeletal gene families are also complexifying elsewhere in Asgard archaea. So it could be that the process of complexification with regards to the cytoskeleton actually occurs before we split off from Asgard archaea. And so this is sort of consistent with that finding. So the cytoskeleton appears to be early. In terms of the nucleus and its genomic organization, I had no idea how to structure this slide, I'm afraid. But I just wanted to point out we have 2 entire supplementary figures just on nuclear functions, chromatin, splicing, RNA and DNA polymerase complexes. But the point I kind of want to make is that on our timeline, we have this split from archaea and the split from alpha proteobacteria from the mitochondria, but most of the stuff seems to be happening even before the split from alpha proteobacteria occurs. So the gut feeling is that cytoskeleton is early. The development of the nucleus as a compartment, and certainly other aspects of genomic biology which make eukaryotes unique, probably happened fairly early on. In terms of the endomembrane system, so I recycled this slide from earlier, but we seem to have a pattern of duplications which indicate the existence of the endoplasmic reticulum as a compartment and the Golgi as a compartment before we have the endolysosomal system being patterned as a compartment. This is data based on archaeal descended genes, and so at the very top end of this, we're having these compartments patterned solely by the archaeal host, that the alpha proteobacteria was not required or essential for patterning the ER and the Golgi. In terms of the endolysosomal system, what we have is this nice correlation between archaeal proteins that tell you the compartment is there and bacterial genes giving function to those compartments agreeing on the same age. So our gut feeling here is that ER and Golgi earlier, the endolysosomal system later, and as those compartments are patterned, they are being patterned with the gene families that give them their present-day function. So the endolysosomal system is being patterned as a digestive compartment at the beginning of its conception. I promised myself I'd give like a little intermediate point before getting to the mitochondria. But what we seem to have is, and remember, my method is blind to horizontal gene transfer, except horizontal gene transfer events which undergo duplication. So I can only see duplications. But it is interesting to consider that we have the patterning of the endolysosomal system, the ability of a cell to endocytose or phagocytose external material, and this sudden increase in bacterial origin duplications, which we posit in the paper is perhaps a signal of the development of endocytosis or perhaps phagocytosis. And finally, the mitochondria, the one that literally everyone cares about, which is my last slide, I'm afraid, or nearly the last slide, is, and there's a little bit of a tease up to it, because although we have the timeline down from our species tree, we know when the mitochondrial ancestors split from other alpha proteobacteria, there is actually a bit of the tree before the mitochondrial endosymbiosis. And actually, even by all our clever methods, we don't actually have that on our species tree. We have to infer when the endosymbiosis was. And we reckon we have a way of doing this. First, we have some genes of archaeal descent, which duplicate and create mitochondrially localized paralogs. And these, all things being equal, these things should only pattern to the mitochondrial compartment when the mitochondrial compartment exists. Second, we have a number of gene duplications of alpha proteobacterial origin, which are involved in establishing the endosymbiotic dialogue. So here is your protein, pre-protein import machinery, which allows the mitochondria to import proteins from the host cytosol, as well as transporters and metabolic enzymes involved in the endosymbiosis. And these seem to undergo duplication early. All things being equal, bacteria don't like to keep on hold of duplications unless it's strictly necessary. And so this burst of duplications could perhaps be explained by being these genes being transferred to the host nucleus, being in an environment much more permissive of duplications. And so we don't get a precise number. There's no node in any of our analysis which corresponds to the endosymbiosis, but we align it fairly early on. And we say that there's a split from alpha proteobacteria, and the window between that split and the endosymbiosis is very small, in the order of 100 million years. And this timeline of development for an organelle, this 100 million years, 200 million years, is similar to the numbers that we have for, if you remember the rather popular, a couple of years ago, the nitroplast was theorized to have been domesticated within 100 million years. Elsewhere at the University of Bristol, the Pereira group has been able to say that the chloroplast has been domesticated within 100 or 200 million years. So the timeline of development, we don't feel is exceptional, but it would be in line with these other estimates. Anyway, and most importantly, it happens towards the end of our timeline. So going back to our introductory slide, and we make a model out of it. So we, at the end of the paper, just to annoy everyone, we added our own little, there's a lot of different models out there for eukaryogenesis. I think everyone should relax a little, so it ended up as being calm. But it just describes this, most of the other models about how eukaryotes came to be are dealing with events or situations or metabolic dialogues. Our model is solely informed about the appearance of eukaryotic features. And so how does this compare to the other models? Well, the ones that start with the mitochondria are looking a bit shaky, so that's not so great. There's a number of models which propose development of the cytoskeleton early on, and we agree with that, but we have this evidence that perhaps cytoskeleton is being elaborated in Asgard archaea anyway, so that's not entirely surprising. Our model kind of rules against a protracted period of serial endosymbiosis because we'd expect to see bacterial duplications going back a long way before the duplications associated with the mitochondria. So we kind of rule out at least a protracted period of serial endosymbiosis. We supposition that the ability, well, we certainly suppose that the endomembrane system of some form or another was developed before the mitochondrial endosymbiosis, perhaps including phagocytosis. And so our model is very consistent with models which propose phagocytosis as the enabling mechanism or the mechanism which allows the mitochondrial endosymbiosis to come about. How many people here are actually earth scientists in disguise? No one? Oh, okay.
They're just being shy.
Just being shy. The acquisition of the mitochondria does coincide with the GOE, this atmospheric rise in oxygen. And so models proposing that the acquisition of the mitochondria was an adaptation to atmospheric oxygen would be consistent with our methodology, with our results. Anyway, that's basically what we have. In terms of future work, I feel this works along this very long stem. And there's a couple of other long stems out there which are equally feature-rich and quite interesting. And so maybe we can apply that to this. I'm thinking particularly of animals, plants, maybe even looking at LCLU a little bit. What we weren't able to do, but we caught as bycatch, were hundreds and hundreds of genes which have no prokaryotic outgroup, but they have a pre-leaker duplication. Regrettably, our method can't date them, but there are just as many pre-leaker duplications of eukaryotic innovation out there as there are of the ones that we found in the same features that we've identified. So any method that can actually time resolve these should be able to independently verify our results for our approach. And again, we're kind of looking into that. So, acknowledgement slide. Oh, you get out relatively early today. I'd like to thank the other authors. This was a big job, I think I phrased in our press release. I'm here mostly as the person who made the method possible and actually does the molecular biology to actually understand what the all things do. But the philosophy and the deep understanding of the whole eukaryogenesis process required some excellent minds to actually hammer down into the final manuscript. I'd also like to, of course, thank our funding providers. Of course, the Lever Human is probably a big name around here, but they were, of course, very helpful to probably helping us publish something which has no applications whatsoever, which is rather nice. And with that, I would like to hand over to any questions.